and people can join as they come. So I was going to start by introducing our speaker. First, just an administrative note that this is in our focus group format, but it's actually a webinar presentation from Dr. Zekavat, and then I will review our latest knowledge portal release. So it's just not in the webinar Zoom format, and it'll look different. So today we're really happy to have Maryam Zekavat presenting with us. She is and correct me if I get this wrong, but I think you're a second-year resident at Massachusetts Eye and Ear Infirmary and Harvard Ophthalmology, and also a postdoc at the Broad Institute. And we've known Maryam for several years now because she approached us to talk about the need for a knowledge portal for ocular diseases and conditions. And we created that, and her data are now in it, some of her results. And so she's going to present on the science behind that data. And please take it away, Maryam.
Awesome. Sounds good. Thank you so much for the introduction. Let me go ahead and share my slides. Oops, one sec. All right. So you're able to see the slides here?
Yes, that's great.
Yes.
Okay, great. So this is work that I started a couple years ago and was finally published a couple months ago in Science Translational Medicine, connecting retinal layer thicknesses from retinal OCT imaging data with ocular and systemic health and with genomics. And this was done across over 50,000 individuals from the UK Biobank with replication in a separate cohort called the Life Adult Study in Germany. And the co-authors are all listed here. And if you go to the Ocular Disease Knowledge Portal, you'll be able to see all of the results that I'll discuss in this presentation and more. So there were two key questions that this work aimed to address. The first of which was, what information can the retinal layers provide on future ocular and systemic disease risk? And the second question was, what genes influence retinal layer thicknesses? And in order to answer these two questions, we utilized data from 5,000 individuals in the UK Biobank, with replication of our analyses in 6,000 individuals from the Life Adult Study. These individuals all had retinal imaging available, OCT imaging available from Topcon in the UK Biobank, and a different OCT machine called the Heidelberg in the Life Adult Study. And basically, when you have OCT, it's a way to look at the different layers of the retina that you can see here. And the layers of the retina all reflect different types of cells in the retina. So at the bottom of the retina is the choroid, which is the vascular supply to the outer retina, which includes the RPE plus Bruch's membrane. Then there's the photoreceptor layer, and above that are different neurons and interneurons, ganglion cells, that then feed the visual information to the brain. And the first thing we had to do was segment out the different layers of the retina. And here are kind of averages of the thicknesses of each of these layers across the UK Biobank population, with similar averages found in the Life study as well. And once we quantified retinal layer thickness across these different layers, then we had to look at what were the disease and the genomic associations. And so we did cross-phenotype association analyses across prevalent and incident conditions, as well as across quantitative clinical and ocular traits. And then we did a GWAS analysis across common and rare variants, and a downstream analysis from the GWAS as well. And our initial findings showed that aging is associated with thinning of nearly all the layers except for the RPE plus Brooks membrane layer, which thickens around age 60. And that's about the age at which we generally see drusen development, or these kind of lipid lipoprotein rich deposits in the outer retina develop in individuals, which can eventually lead to age-related macular degeneration. And then we looked at two of the main common ocular conditions, AMD, or age-related macular degeneration, and glaucoma. And we saw that, as expected with AMD, or as has been seen in prior observational studies, we see that the fraction of individuals with AMD increases as you get a thicker RPE plus Brooks membrane layer, and as you get a thinner photoreceptor layer, which is indicative of drusen deposits, which are kind of in this Brooks membrane layer, develop in this Brooks membrane layer, as well as photoreceptor degeneration, which both contribute towards macular degeneration. And then we found, as expected, glaucoma is associated with thinning of the inner layers of the retina. So those were the top layers, the ganglion cell layer, the retinal nerve fiber layer, which are associated with glaucoma, where thinning of these is associated with glaucoma. So these were kind of some initial findings. Then we went to look a little more deeper into the ocular associations, and so we did a phenomide scan across all diseases, and this is the results across specifically incident ocular diseases. And this is in a survival model using a Cox proportional hazards model, adjusted for age squared, sex, height, weight, ethnicity, and spherical equivalent. And spherical equivalent is kind of a measure of how myopic you are, or the size of your eyeball, basically. And when you adjust for these factors and look at incident ocular diseases, indeed, there is significant association with glaucoma as well as with macular degeneration. And these are some examples of the hazard ratios, just to give you a sense. The hazard ratios are all reported per standard deviation decrease in the respective layer. So here, every 1 standard deviation decrease in the RNFL thickness was associated with a 2.5-fold increase risk of future primary open-angle glaucoma. And then similarly, every 1 standard deviation decrease in the photoreceptor thickness was associated with a 1.4-fold increased risk of future AMD diagnosis. And then we went to look a little further. So beyond the eye, is there an association with incident mortality? And indeed, when you look across the different retinal layers, thinning of the retinal layers, in particular, thinning of the inner retinal layers that comprise the ganglion cell complex, which is a combination of these top 3 layers here, the retinal nerve fiber layer, the ganglion cell layer, and the inner photoreceptor layer, are all, inner plexiform layer, are all associated with increased risk of incident mortality. And photoreceptor thinning is also associated with increased risk of incident mortality after adjusting for the factors listed here. And then kind of looking a little further in that binning by age, you see here in the Kaplan-Meier curve that across different age bins, individuals with either a thin photoreceptor or a thin ganglion cell complex consistently have higher risk for mortality compared to those with a thin photoreceptor and thin GCC layer. And so the question next was why? Why is there an association with incident mortality? And to look at that, we looked across the other phenotypes in the PheWAS. And so these are some of the associations by system. So now looking at incident cardiovascular disease, we see that there's significant associations between the ganglion cell complex layers as well as the photoreceptor layer thinning and multiple cardiovascular outcomes, including hypertension, with every 1 standard deviation decrease in the photoreceptor segment layer being associated with a 1.25-fold increased risk of congestive heart failure, as well as a 1.5-fold increased risk of abdominal aortic aneurysm. And then there's also significant associations with other quantitative clinical traits like heart rate and blood pressure, BMI, and percent body fat. And then now looking across incident pulmonary diseases, we identified significant associations between, again, the same group, so the ganglion cell complex group and the photoreceptor layer, with increased risk of future diagnosis of pulmonary disease. And this includes all the conditions listed here. But kind of the main branch or the most significant association was with COPD or obstructive pulmonary diseases, with every 1 standard deviation decrease in the photoreceptor segment layer being associated with a 1.5-fold increased risk of future diagnosis of COPD, suggesting that, again, like preclinical disease as seen as manifested in your eye is suggestive of a future diagnosis of, in this case, pulmonary disease. And indeed, there was significant association with quantitative traits of the lung as well. And this is showing FVC and FEV1 from pulmonary spirometry tests. So this is where you have a patient breathing into the spirometer and you measure how much the spirometer level moves as they breathe in for 1 second here, forced expiratory volume in 1 second, and the vital capacity as well. And you can see that with every standard deviation decrease in photoreceptor thickness, the FEV1 and FVC are decreasing as well. In other words, poor pulmonary function is associated with photoreceptor thinning, in addition to thinning of the GCC layer as well. And these tests were all performed, the quantitative tests were at the same time of retinal imaging. So it was as the same baseline visit. And now looking at renal conditions, we identified that photoreceptor thinning is also associated with multiple renal conditions, as well as their counterpart quantitative traits. There's association with chronic kidney disease with every 1 standard deviation thinning of the photoreceptor layer being associated with a 1.2-fold increased risk of chronic renal failure, and associations with creatinine and urea, which are markers of kidney function from the blood as well. And then there were also associations with incident neurologic and psychologic conditions. Here we identified that across incident neurologic conditions, the inner layers of the retina, or the ganglion cell layer, is associated with multiple sclerosis and epilepsy, with every 1 standard deviation thinning of the ganglion cell layer being associated with a 2.8-fold increased risk of future multiple sclerosis diagnosis. And then there were also associations with psychological diseases, including alcoholism, alcohol-related disorders, delirium with ganglion cell layer, and with schizophrenia with the photoreceptor layer thinning, with every standard deviation thinning of the photoreceptor layer being associated with 1.9-fold increased risk of schizophrenia, of future diagnosis of schizophrenia. So those were all the phenotypic associations. Next, we did a genome-wide association study that did a GWAS across each of the 9 different layers, and that identified 259 inherited loci associated with retinal layer thicknesses. And this is kind of looking at the number of loci per layer and its respective genetic heritability. This plot here shows you that there's a lot of layers which have a shared locus. And so, for example, the black layer here, the black region, the black locus here, or the ACTG1 locus is associated in 8 out of the 9 layers, is genome-wide significant in 8 out of the 9 layers. And then similarly, the num locus here is in 7 out of the 9 layers, and the pink MEF2C locus is in 6 out of the 9 loci. And then the gray loci are unique to a specific layer, and so those are all listed here. We were able to successfully replicate the genome-wide association study in a separate cohort of 6,000 individuals from Germany that had a different type of imaging and were separate from the UK Biobank. It's Heidelberg instead of Topcon camera was used to take a photo of the retina here. And again, here, significant correlations between the UK Biobank beta versus the Life Adult Study beta for the variants that were significant in the UK Biobank. And we did do some downstream analyses to look at after the GWAS associations, we did kind of gene prioritization using Hilary Finucane's POPs algorithm across each of the loci and each of the layers, and then use that towards pathway enrichment analysis, which identified significant enrichment of pathways associated with cellular death and senescence and angiogenesis, as well as neuron-related pathways and different melanosis-related pathways as well. Melanin is a big factor in the retinal pigment epithelium, as it's so termed. There's the term pigment in it, and different levels of melanin can affect retinal layer thickness, I guess, as well. And so after doing all those post-GWAS analyses, we kind of took a more holistic and zoom out approach and said, okay, now is there some correspondence that we can identify between the epidemiologic analyses that I presented earlier and the genetic associations that we're seeing? And so we did comparative epidemiological and genetic associations, looking at the relationship between the epidemiological associations of any phenotype X and outcome Y, as well as genetic associations of that same phenotype X and outcome Y using polygenic risk scores for X. And this analysis was done across, limited to the quantitative phenotypes that we did a PheWAS across. We developed the polygenic risk scores ourselves across all the quantitative phenotypes, and then looked at the comparative epidemiological link between X and Y. And this was done in order to kind of simulate many Mendelian randomization analyses, although it wasn't a formal Mendelian randomization analysis, kind of across a broader range of about 100 traits. So this analysis was able to link a thinner photoreceptor layer with risk of AMD, and indeed we found that when you look across epidemiologically and genetically, a thinner epidemiologic photoreceptor layer as well as thinner polygenic photoreceptor thickness score were both associated with increased risk of AMD. And we were able to link a thinner RNFL layer with glaucoma. And indeed, you can see that epidemiologically, a thinner RNFL layer is associated with glaucoma, but also genetically, individuals who have a lower polygenic risk score for RNFL thickness also have increased risk of glaucoma. And so that kind of looked at the connection between thinner retinal layers and ocular phenotypes, but we also looked at the relationship between ocular and systemic phenotypes and retinal layer thickness. And we identified all the associations listed here as having consistent epidemiological and polygenic risk score associations between the phenotypes and retinal layer thickness. And I'll go through a few examples next. So this is showing an example linking lower pulmonary function with a thinner photoreceptor layer. So again, we can see epidemiologically that a lower FEV1 is associated with a thinner photoreceptor thickness, but also genetically, a lower FEV1 polygenic risk score is also associated with a thinner photoreceptor layer. And then similarly, a higher systolic blood pressure, both epidemiologically and in a polygenic risk score, is associated with thinner GCC and RNFL layers. And again, these are like crude plots here, but this analysis for the results listed here was all after adjustment for the prior aforementioned covariates. And so just to conclude, this was one of the first population-wide OCT retinal layer thickness, phenome and genome-wide analyses across 45,000 UK biobank participants with replication in 6,000 live adult study participants. In the phenome-wide association analyses, we identified that thinner inner and outer retinal layers are associated with multiple ocular, cardiovascular, pulmonary, and neurologic and psychological conditions. And then in the genome-wide association, we identified 259 common loci, and I didn't show it in this presentation, but we also did an analysis across the exome data that identified rare disruptive variants across a burden analysis from 11 different genes associated with retinal layer thickness. And then comparative phenotypic and genotypic associations identified multiple putative causal links between ocular phenotypes and retinal layer thickness, as well as systemic phenotypes and retinal layer thickness. So I want to thank all the individuals involved with this work, in particular my co-first author, Saman Durgar, and individuals involved at Mass Pioneer, Dr. Zabardast and Sayuri Sekimitsu, and at the Broad Institute in particular, Pradeep Natarajan, and co-authors from both labs, as well as the life adult study that was involved with replication. And then that gets me to the Ocular Knowledge Portal, which is the topic of this presentation. And this is kind of showing the Ocular Knowledge Portal. This is our specific paper that's discussed. And then we can see the genetic analyses, and this is showing a preview of what the genetic analyses looks like for the photoreceptor retinal layer thickness. We can see all of the top SNPs and indels associated, as well as the gene level associations, and the top pathways for photoreceptor retinal layer thickness, which include vascular endothelial cell proliferation and macular dystrophy. And we can look at the genetic associations and correlations and the enriched annotations. And this is showing a little bit of a more unique analysis here that we did, which was the cross-phenotype association. This is looking at the prevalent diseases. I think this is still looking at the photoreceptor layer thickness, and you can see all the phenotypic associations grouped by disease category. you can look up your favorite disease, you can look up your favorite retinal layer and its associations, both the prevalent and here you can see the incident results. And in this one, you see associations with ischemic heart disease, or their cardiovascular outcomes, etc. You see associations coming up with sleep apnea for the photoreceptor layer as well, which is one of the pulmonary conditions that we see come up. And there's the quantitative traits and blood biomarkers that we looked at. And this is looking at the choroid thickness and all the associations here. And that pretty much concludes my talk. Happy to answer any questions.
Hello, can I have a few questions, please?
Yeah, go for it.
It's a really nice talk. It's great work, and I have a few questions. First is the retinal layer thickness data from OCT imaging data in the body is publicly available, or if not now, when?
I think a lot of the retinal layer thicknesses have become publicly available, like it's available through the UK Biobank for download. Some of the ones here have not been made available yet. I think it's the photoreceptor layer thickness, and a few of the smaller layers have not been made available yet, but I believe there is a plan for upload onto the UK Biobank portal for those as well.
Okay, thank you. So we know that UK Biobank has proteomics data among 50,000 subjects. Just curious, among those also around 50K UK Biobank subjects with OCT data, how many have proteomics?
How many have proteomics? Is that what you asked?
Yes, only
I actually
proteomics.
don't know. I think the proteomics it came when like after I started this project, so I didn't look into that, but I think that's a good avenue for future work.
Sure. And among those 50K subjects, how many have AMD and how many have a glaucoma? And how those two phenotypes being defined in clinical?
I couldn't quite catch that. How many have what and what?
How many have AMD and how many have a glaucoma?
Oh, AMD, you
Yes.
said. Okay. That's all in the paper. I don't know it offhand, but it's a very small proportion. I think it's about 1,000 people have AMD, and a few the population with glaucoma is more. Of course, it's more common, but I don't remember offhand.
Okay, thank you.
Yeah.
Thank you so much.
Thanks so much, Maryam. Other questions from anyone? Well, I have one, but I also wanted to say, there's an old quote, the eyes are the window to the soul, but I think maybe you've shown that the eyes are the window to the body.
Yeah.
And this is very speculative, but do you ever see this like being used for prediction of disease risk, just looking at someone's eye thickness, layer thickness?
Yeah, that's a great question. And I think that's kind of the next step from here is to see if we can predict disease risk using retinal imaging data. And I would probably at that point combine both OCT and fundus photos together for.
Not
I
sure.
think Maryam may have frozen.
I wasn't sure if it was me or Maryam.
No.
See if she comes back. Okay. Well, let's see. There, okay. She left. So, okay. I think we'll just go on with the webinar. If she comes back and has more to say, we'll let her say it. Okay. Oh, wait a minute. Is she coming back? There she is. Hi.
I think my internet got disconnected for a moment.
Yeah. So you were just saying about the predictive value of these measurements, possible predictive value.
Yeah, we haven't looked into that yet, machine learning to directly predict future events, but I think that would be an interesting next future avenue.
Yeah, great. All right. Well, thank you so much. I'm really happy you could come and talk
Yeah,
to
thank you.
me. Okay. I'm going to go on to the next part of the webinar then.
All right, thanks everyone.
Take care. Bye-bye.
Bye.
So let me share my screen. Yeah, I was going to show you the ocular knowledge portal in case Maryam didn't, but she gave us a good demonstration of her data. And I'll just show you briefly that there are lots of other data sets here as well. Not only for ocular phenotypes, but for other phenotypes that could be relevant, such as cardiovascular phenotypes. And this is the ocular knowledge portal is under the umbrella of our knowledge portal. So it has, a subset of the data sets that you'll find in the association to function knowledge portal. So let's see, go to my PowerPoint. I wanted to now just give a really brief rundown of our latest release. We had a release last month. Oh, let's see. Mackenzie, are you seeing the correct screen here?
Yes, I'm seeing your slides.
Oh, good. Okay. Okay, so we had a release and we haven't really publicized it much, so I just wanted to give you an overview. And first of all, we have some new features in the portal itself, and so I wanted to demonstrate those. Yeah, actually, I'm going to get out of the PowerPoint right now and go back to the portal to show you that. Okay, so one of the new features is a whole new page. And let's see, so it's available in all the portals. I guess I'll go to the Association to Function Knowledge portal to demonstrate it. So this page, as you know, we have core pages that are focused on a gene or a variant or a region or a phenotype. And now we've added a tissue page. So you can put a tissue into the homepage search box. And we'll just do eye since we've been talking about eyes. And so this, I want to stress, this page is a prototype. It's not, it will have a lot more information on it, and some of the aspects of it don't quite work as they should yet, but we're working on it, and we just wanted to get it out there and get some feedback on it. There is a full documentation page that you can go to, which explains each section and tells the sources of the data and how to use the navigation and so forth. So this page is about the eye, and the first display here at the top shows us.
Maria,
Yes.
right now I see your spreadsheet and
Oh no, you're seeing
yeah,
my.
so I think it changed.
Okay, let's try that again.
Sorry.
Good thing I didn't have the news up there.
No
Let's
worries.
see. Desktop one. Okay, yeah.
It was fine, and then I think it switched, so
Okay.
that looks great.
Thank you. Okay, so yeah, so you didn't see when I clicked on this documentation link that we have a whole documentation page here, and that's easily accessible. Okay, so then this display shows the top genes that are expressed in this tissue, or the genes that are most highly expressed in this tissue. And we have these nice violin plots for each of them. When you mouse over, you can see points that are in individual data sets. And the highlighted points here are all in the same data set. You can adjust the view by minimum sample count, or you can change the scale. Right now, the only collection that we have is all these data are from GTEx, but in the future, we'll be adding more collections from different resources. And then you'll be able to select them here. So here we have some of the transcript per million values for these genes. And then if you want to look at the individual data sets, you can click this button, and then you will see, first of all, the detailed biosample that the data set comes from. So this one was retina. There's a link to the data set at CMDGA, and then all those TPM values and the number of samples and so forth. This, you can page through this display. It's not only the top 10 genes, there are many genes, and you could just click through the pages to look at other genes. The second part of this page is global enrichment. So what this is, it looks at genetic associations for all of the phenotypes that we have, and it looks at whether those associations are enriched within regions annotated as these different types of regulatory elements in eye tissue. So for example, here, first, I don't understand why the biological reason for this, if there is one, but genetic associations for height are significantly enriched within enhancer elements that are active in the eye. I can't really explain that one. Maybe because height has so many genetic associations. There are thousands of them. I think it's the phenotype that has the most. So maybe that's the reason for that. But then you can also see, for example, some blood pressure associations are enriched, and that makes sense because obviously blood pressure is important for eye function. So this is our tissue page. And please check it out and give us feedback. There's a lot more tissue-specific information that could go here, and we'd like to know what you think. So let's go back to the home page. One quick thing I wanted to mention is that we've changed our display a little bit of some of our statistics. So we used to have graphs, but we're getting so many data sets and phenotypes now that the graphs were getting very crowded. So now we summarize some stats about what's in the portals here. And we are looking into adding more and better summaries on individual pages, maybe, so you can see, exactly what the summarized results that you're seeing in the portals have come from. Another change we've made is to the phenotype page. So let's look at the phenotype page for. Oh, sorry, I was going to the gene page. Let's go to the gene page first. So we'll look at the. Wait a minute. I'm sorry, I'm confusing myself. I did want to go to the phenotype page. We'll go to the BMI phenotype page. Okay. And another pretty simple addition that we have made to this page is that if you scroll down here, we previously had this display that has, it had the common variant gene level associations for the phenotype of the page. But now we've added tabs to this. So in addition, you can see the huge scores and rare variant associations. So the huge score combines common and rare variant associations to make a prediction about the likelihood that a gene is involved in a phenotype. And so this tells you all the genes that are predicted to be significantly involved in the body mass index. Then the rare variant associations, again, these are gene burden tests done on the rare variants that are within the coding sequences of these genes found by exome sequencing. And again, you can see that the top gene is MC4R here. And so finally, another new thing that you can see is on the gene page, and I just clicked on MC4R to go there. Excuse me. And that is in the gene expression section. Now here is a similar violin plot. This is tissue-specific expression for the MC4R gene. And something that we've added is annotations that for experiments that were done in disease state. So we only have a few diseases so far, but we'll be adding more, and we have some pretty important cardiometabolic diseases. So here, if we want to look at data that were collected in acute kidney failure, we've filtered that down and we can see it. So there's not, okay, this is not a great example because this gene in the kidney, in acute kidney failure appears to be not expressed, but there's more information for other genes. So this is, we're going to be adding to this feature, but it's just a way of looking at differential expression for a gene in health and disease. Okay, so then briefly, I just wanted to show you, go back to the PowerPoint and show you the new data that we've added. Let's see. Now, Mackenzie, I'm guessing that you're seeing the presenter view?
Yes, sorry, I was on mute, but yes,
That's
this is
okay.
pretty much a view.
This is there. Okay,
Now we're good.
good. Okay. Okay, so as usual, we've added a lot of genetic association data sets. We're adding more and more these days. Our colleagues at EBI GWAS Catalog are helping us to get and harmonize data sets. And so with this latest release, we added 72 covering 134 traits. And this is just their distribution over different categories of phenotypes. So currently in the Association of Function Knowledge portal, which has everything, we have almost 800 data sets covering more than 900 phenotypes. Some of the highlights of those for anyone who's interested in particular phenotypes, we've got some very large data sets for the cardiovascular and cerebrovascular system with over 1 million subjects for abdominal aortic aneurysm, GWAS, and atrial fibrillation GWAS. We have a lot of other more granular cardiac phenotypes. We also have some neurological and nervous system phenotypes, for example, for Parkinson's and Alzheimer's, and biomarkers of neurodegenerative disease. We've added, as I think we've talked about here before, we've added a new aging and longevity portal, and we've added some aging GWAS to that in this release. We've added a lot more with the previous release, and including a really huge GWAS on aging-related traits. We have some nice musculoskeletal data sets. There's a large one for a forearm fracture and an interesting one for skeletal proportions that looks at all different measurements of how long certain bones in the body are. We've added some GWAS for the kidneys, for pediatric steroid-sensitive nephrotic syndrome, kidney stones, also some reproductive systems for preterm and postterm birth and birth weight, things like that. Also some sleep phenotypes, GWAS for REM disorder, and chronotype, narcolepsy, etc. And finally, also some respiratory GWAS, and of course, we wouldn't neglect the endocrine system. We have a couple from the MAGIC consortium on insulin levels and random glucose. We have one from the Prodigy consortium on a youth onset T2D, and then two whole genome sequencing analyses from TopMed and all of us. Then, yes, we've added more ocular phenotypes as well in this last release, and digestive system is something we haven't really had a lot of data for yet, and there actually aren't many GWAS data sets, but we're trying to collect some interesting ones, so we've added some with this release. Now for genomic annotations that come to us via CMDGA, this graph just represents everything that's currently in there, not only the previous release. Our release schedule and CMDGAs aren't exactly synchronized, so it's easier to represent it this way. All of these annotations are currently represented in the knowledge portals, and in contrast to the genetic association data sets, when you look at any of the knowledge portals, all of these annotations will be accessible. So that is a difference. Genetic associations are segregated by phenotypes in the portals, but these are all available. So there are lots of different types of annotations, such as target gene predictions, chromatin state, and so forth. And then they're also developed, distributed pretty nicely over tissue categories. Blood is still the most common one, since of course blood is an easy tissue to sample, but there's a good number of samples for other tissue types as well. And yeah, that is it for my presentation. Are there any questions about anything? Oh, I see some things in the chat. Let's see. Yunji, are you mistaken that eye expression data is not available in GTEx? I thought it was, but I am not totally sure. We can take a quick look at GTEx, I guess. Let's see. Oh, the person left the webinar. Okay, well then I guess we don't need to. I'm pretty sure that it is in GTEx. Okay, all right. Well, If there are no further questions, thanks everyone for coming, and we'll see you next time. Bye-bye.
Thank you.